Hello and welcome to the November Longevity Review, where we will take a look at how 16-8 time-restricted eating in healthy, normal-weight individuals can still reduce weight and fat mass, as well as improve their immune system and make their gut microbiome younger. How ergothioneine could improve cognition in older adults with mild cognitive impairment. And for the first time, two Canadian content studies, the first of which looks at how senolytics could help with multiple sclerosis, while the second investigates how Pterostilbene targets the hallmarks of aging. This review is presented to you by the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do, then please check out the links below this video, where you can also find links to the newsletter and studies reviewed here. And lastly, don't forget to like this video and subscribe to our channel. So let's take a look at the first study. This study looked at time-restricted eating, a form of intermittent fasting where you only eat during certain hours of the day. It has shown numerous benefits and could potentially increase your longevity. The most common form is called 16-8 and it involves fasting for 16 hours and only eating during 8. And that is the form this study looks at. So let's start by taking a look at the graphical abstract. And as you can see here on the top left, the participants fasted between 5 p.m. and 9 a.m. for 16 hours and ate between 9 a.m. and 5 p.m., having 3 meals during that time. There were 49 participants in total and also 10 controls who were not following this protocol. 90% of the participants were female, and the study was conducted in China. The participants had all their food provided for them in a canteen. It was well balanced and calorie matched for their weight and activity profile so that they didn't have a calorie deficit. The participants were all of normal weight with a BMI of under 25. They were followed for 30 days and their weight, peripheral blood, and gut microbiome were analyzed. Over the course of the study, most participants saw their body weight and body fat decrease, and their body moisture and skeletal muscle rate increase. I believe that skeletal muscle rate refers to their metabolism. Their immune cell profile changed in a potentially beneficial manner. You can see various immune cells either decreased or increased. But more importantly, their senescent CD4 plus T cells, which are a marker of senescence and increase with age, which we don't want to have happen, decreased over the course of the study. You can sort of see in this graph here, maybe not too clearly, but the levels of these cells decrease, which is a good thing. Additionally, the TCRs and BCRs, which stand for T cell receptors and B cell receptors, changed as well. The authors state that the significantly reduced large clones of heavy kappa and lambda BCR chains showed that time-restricted eating was able to improve immune system function in healthy individuals. Now, I'm not an expert in the immune system, so I will take the authors by their word. Next, looking at the metabolites in the blood, the age-associated ones declined and anti-inflammatory ones increased, which is also a good thing. Lastly, looking at the intestinal microbiome, microbial diversity increased, as did the probiotics. All of this indicates positive aspects to this diet. Now let's take a look at this study in a little bit more detail, starting with the changes in weight and fat. Once again, in the top left, we have the study design. And in the top right, it shows the percentage of participants who lost weight. And that was 47 out of 49, so 95.9% of them lost weight. And here in C is the percentage breakdown of those who lost weight, from less than 1% weight loss to over 5% at the bottom here. Now, it looks like they made a mistake with one of the colors here. However, out of the 47 who lost weight, 8 lost less than 1%, 4 lost between 1 and 2, 12 lost between 2 and 3%, so that's the wrong color. 12 also lost between 3 and 4, 5 lost between 4 and 5, and 6 lost over 5% of their body weight. And as you can see in D here on the right, they started off losing a fair bit of weight that sort of leveled off. However, they continued to lose weight throughout the course of the study. And as their calories were matched to their body weight and activity levels, the thought is that this weight loss was due to this eating pattern. And as we can see in E here, body fat rate, BMI, basal metabolism, and fat weight decreased for most of the participants between day 0 and day 30. So this reached statistical significance, as can be seen by these p-values on the top of each of these charts, with a reminder that a p-value of 0.05 and below is statistically significant. On the right here, we can see muscle mass, visceral fat, body moisture and skeletal muscle rate, which again I believe is about the muscle metabolism. And one unfortunate aspect of intermittent fasting or fasting in general is that not only do you lose fat mass, but also you can lose lean muscle mass. So it's important to also do some resistance training at the same time to try to keep that muscle mass up. Now, body moisture supposedly decreases with age and obesity, so raising it is a good thing, which is what happened to the participants here. And once again, all four of these reached statistical significance. Next, they looked at the immune cells, and we already mentioned the CD4 plus senescent T cells in the graphical abstract, and that figure can be seen here in the bottom right. We won't go into the details of this as the immune system is not exactly my strong point, and that was by far the most significant finding. It's similar with the next figure, which again shows those positive changes in the BCR chains. And moving swiftly to the metabolites, there were a large number of changes between day 0 and day 30. If you can see over here, total metabolites, 1,308, 202 were downregulated and 219 were upregulated. But I'm just going to focus on G briefly. And all of these metabolites from PA to L-serine are anti-inflammatory. And as you can see, they were all upregulated. And the only beneficial one, which was downregulated, was 5-methylthioadenosine, or something like that. Anyway, as you can see, there were lots of changes, and the authors state that most of these changes were in a positive direction in terms of health and longevity. Lastly, the authors looked at the microbiome, which according to the Chow-1 index here on the top left, showed that there was an increase in diversity, though according to the Shannon index, it didn't reach statistical significance. And in terms of changes to the major phyla, the firmicutes and proteobacteria decreased, going from 86 to 73 and 9 to 6%. And the bacteroidetes and actinobacteria increased, going from 2.3% to 3.4 and 2.2 to 16.9. Now, just briefly, the firmicutes, although they are generally beneficial, they can also lead to weight gain, and the proteobacteria are generally seen as harmful, whilst the bacteroidetes and actinobacteria are seen to be beneficial, with the bacteroidetes producing short-chain fatty acids, which in our intestine, which is again, a good thing for us. Now moving to the genus level, which is just above species, so species are all sort of part of a genus. The Akkermansia and Rickenolacea were the only two which showed a statistically significant change, both of them increasing, and both of them are beneficial and associated with longevity. Now, Akkermansia is anti-inflammatory, is good for obesity, glucose tolerance, and, well, they say longevity. Rickenolacea are more often found in lean individuals, supposedly. So both of these increase. And actually, I've seen a number of studies where Akkermansia have been given to mice or what have you, and they have shown to be beneficial. So it's good to see that they have increased. And in fact, you can get supplements containing them. Overall, the researchers say that the gut microbiome profile of the participants improved and move towards that as seen in centenarians and younger people. So this was beneficial. Taken all together, as we return to the graphical abstract here, 16-8 time-restricted eating proved to be beneficial to the participants, leading to weight loss and fat loss, improving their immune system and metabolic profile, reducing their senescent CD4 T cells, and making their gut microbiome more youthful. In addition, the participants subjectively reported that they had greater gastrointestinal comfort, improved mood, and improved mental outlook. So they just seem to be generally happier. I guess if an intervention can make you happy while also improving various longevity aspects, then that's a good thing. On to the next study. In this very small study from Singapore, researchers gave adults over the age of 60 with mild cognitive impairment ergothioneine supplements to take to see how it affected their cognition. Ergothioneine is the main active ingredient in mushrooms and is a very powerful antioxidant. It has been likened to a vitamin, and epidemiology studies have shown that it can reduce mortality. Prior research by these same researchers has shown that people with mild cognitive impairment and dementia have lower levels of ergothioneine. Also, cognitively normal adults with lower levels were shown to have faster cognitive decline over 5 years. The researchers therefore wanted to find out what would happen if they gave these older adults 25 milligrams of ergothioneine 3 times a week for a year. Now, what stood out for this study for me, apart from the fact that I love mushrooms, was the fact that this study lasted for a year. Most studies in supplements are only for a few weeks or at best a few months, so it was good to see a longer-term study done. Let's start by taking a look at the study design. Due to the fact that the study took place during COVID, the researchers couldn't recruit the number of people that they wanted to. Hence, they are calling this a pilot study. But it was randomized, double-blinded, and placebo-controlled. As mentioned before, the participants were given 25 milligrams of ergothioneine to take 3 times a week for 1 year. They visited the clinic 14 times over the course of that year, once every 4 weeks. Their blood was collected 5 times over the course of the study, and they completed 3 cognitive tests before, in the middle, and at the end of the study. Looking at the baseline levels of ergothioneine down here, we can see that there was no correlation between their initial mini mental state examination here on the left and the levels of ergothioneine, which is represented by ET, maybe the slightly sloping line, the worse their mental state was, but it did not reach statistical significance, nor was there a correlation with age. You can see from 55 to 85 here, again, levels of ergothioneine, and although the line is sloping slightly upwards, it had a p-value of 0.53, which was just above the threshold, the threshold for it to reach statistical significance. And also, when broken down by gender, there was no real difference between men and women. And hercynine and S-methyl ergothioneine are 2 metabolites of ergothioneine, and also they showed no difference between men and women. Now let's see what happened once they took the supplement. And here we go. So on the top left here in A, you can see this is ET ergothioneine from week 1, or the first visit, visit 7 in the middle, visit 14 at the end. Red is control, blue are the treated patients, and you can see that levels of ergothioneine rose consistently over the course of the year, showing no signs of leveling off, and it reached quite considerable statistical significance by the end. In fact, it reached it also at the middle of the study. And the same goes with hercynine and S-methyl ergothioneine, again, 2 metabolites. The levels rose and reached statistical significance over the course of the study. So we can see that there weren't any bioavailability issues. Next, the researchers reported on the safety profile. And won't go into the details here, but according to this table and what the authors say, that all the various blood metrics from total white blood cell count all the way down to hematocrit and platelets show that these levels stayed within the normal range. So ergothioneine was shown to be safe for the participants. However, let's take a look at the results for the cognitive tests. For the first one here, the RAVLT, which stands for the Ray Auditory Verbal Learning Test, which measures learning ability and memory. You can see that the blue line, which again is the treated participants, rose during the time for both the immediate recall and delayed recall. It's behind this open sign, and that increase reached statistical significance. For the placebo patients, there was no real change. And just like the levels of ergothioneine, there appeared to be a continuous improvement here. However, for the other cognitive tests, such as digit, CTT1, BDT, and semantic fluency, there was no real change during the trial, and there was no real difference from the placebo group. So although only one of these measures improved, the authors nevertheless thought that it was a significant improvement and an indicator that ergothioneine might improve memory and learning. Lastly, they looked at plasma biomarkers. And the first one here on the left, NFL, that stands for neurofilament light chain, which is a common marker for neurodegeneration and brain damage. In fact, in traumatic brain injuries, it rises considerably. And as you can see here, while the treated participants, the levels stayed the same and might even have dropped a little bit towards the end of the study, for the placebo group, it rose throughout this entire time. Now remember, of course, the participants had mild cognitive impairment, so we would expect to see NFL levels to rise. For other plasma biomarkers, such as brain-derived neurotrophic factor, TNF-A, and IL-18, which are markers of inflammation, and protein carbonyls, which is an index of oxidative protein damage. So although these levels didn't change, since the blood was taken from the periphery, they wondered if these markers might have actually improved in the brain itself. But the fact that NFL didn't rise in the treated group is a major positive. The researchers do say that larger studies are needed, and hopefully they will be carrying them out. However, the main takeaway of this study was that ergothioneine proved to be safe and showed tentative signs of improving cognition and reducing damage in the brain. And I guess it wouldn't do you any harm to eat more mushrooms. They certainly are delicious, at least I think so. On to the first Canadian content study. I was very excited when I came across this paper, as it is the first Senolytic study coming out of Canada, or at least I think so. In fact, if you know of any other studies, please mention them in the comment section below or send me a direct message. This study from the University of McGill looked at how senescent cells and senolytics might impact multiple sclerosis, which is an autoimmune disease which attacks the central nervous system. The authors wanted to see if senescent microglia worsen the disease and if their removal could improve things. Microglia are immune cells in the brain which have a good and a bad side. They clear out pathogens and remove debris. However, they can also cause inflammation and promote tissue injury. Now, it says experimental autoimmune encephalomyelitis in the title here. That is because they used a mouse model which has experimental autoimmune encephalomyelitis as a substitute for multiple sclerosis. It is used to study the disease in mice. Firstly, the authors looked at the differences in the microglia between normal and diseased mice. So let's take a look at what they found. So first you can see in figure 1a here on the left, they did RNA sequencing of single cells. Here's the RNA difference between diseased and normal control mice. So the diseased mice are in blue here with EAE, which again stands for experimental autoimmune encephalomyelitis. And there is not much overlap between the blue and the red, showing that there were some stark differences. Next, they looked at the pathway differences, and on this chart, further to the right means a greater impact, and the top 5 ones here are, you can see, pathways of neurodegeneration, multiple diseases, Alzheimer's disease, amyotrophic lateral sclerosis, Parkinson's disease, Huntington disease. So these were clearly diseased microglia. Now, of greater importance to us is G here, which shows the microglia expressing these markers. The first one here being BCL2L1, which is a marker of senescence. And here is the percentage of the microglia which were expressing that. And you can see here that BCL2L1 was expressed in over 20% of the microglia of the diseased animals, indicating that they had a high percentage of senescent cells. Next, they looked at microglia taken from multiple sclerosis patients. And without going into the detail here, on C and F is what we're going to look at. And here you can see control, chronic inactive, and chronic active, because one aspect of multiple sclerosis is that it can flare up and then sort of subside for a while. And when it flares up, that's called chronic active. And you can see that when it is active, BCL2L1 is expressed quite strongly. The same goes with F here, which is simply from a different data set. You can see chronic active here in BCL2L1. They have such snappy names, don't they? They also stain the cells taken from the human patients. And I mean, it's hard to tell from these pictures of cross sections, or I guess just staining of the cells. But according to the authors, these cells once again showed an increase in this BCL senescent cells, or at least they showed up in the staining. Hard for me to really tell from this, but that's what the text says. So it is clear that these senescent microglia cells are present in multiple sclerosis patients. Next, let's see what happened when they applied senolytics to the mouse model. So first on the left here in A is the study design. And firstly, they induced the disease in the animals, and then they were monitored from 7 days onwards. And as soon as they showed signs of disease onset, they were given the senolytic. Now, the senolytic is called navitoclax, or ABT263, and I'll call it navitoclax from now on. Navitoclax was given because it targets that BCL senescent marker, and they were given it daily until day 18, at which point they were sacrificed and examined. Now, figures B to I simply show how they sort of sorted out the cells from the spinal cord to isolate the cells they were looking for. Once they had done that, they characterized the various immune cells and compared them to the control animals. The graphs J to Q here show various different immune cells, and the only ones that showed a significant decrease or significant change, and that is shown by the star up here, which shows that it reached statistical significance. And out of these 8 different types of immune cells, the only 3 which had a statistically significant change, so J, M, and N down here, were ones harboring the sort of senescent microglia. So this was a positive indication that the drug was only targeting those cells which they were after. The other ones, although they might appear to show a change, none of these changes reach statistical significance. And just to clarify, in case you hadn't noticed it yourself, the gray bars are the control animals and the light blue bars are the treated animals with navitoclax. Now that we've seen that they targeted the correct cells, let's see what effect it had on the animals. So once again, starting with the study design here on the left, the animals were again induced to have this disease, and once they started showing signs of the disease, they were started their treatment. At the peak of the flare-up, they were tested for vision and motor skills before once again being sacrificed at day 18. Now in B here, you can see at the bottom days post treatment start, so days 1 to 5. And the score here is for the severity of the motor skill loss in multiple sclerosis or experimental autoimmune encephalomyelitis. And the black line represents the control animals, and the light blue line represents the treated animals. And as you can see, treatment started when the first symptoms appeared, and at the beginning they were pretty much the same. And indeed on day 1, there was no real difference between the treated and control animals. However, after that, there was a major divergence, and the treated animals had much less worsening than the control animals, and this reached statistical significance, again, as shown by these stars, and 2 stars means it's of even greater statistical significance. And in C here, they looked at the weight of the animals, and as you can see that once the treatment started, the treated animals maintained their weight or improved their weight much more than the control animals who lost a fair bit of weight. And on the right here in D is the optomotor response. So I guess there's sort of vision fundamentally, and blue indicates sort of no response or a 0.3 response, and red indicates a 0.35 to 0.4 response, which I guess is better. And you can see for the vehicle or the control animals compared to the treated animals down below, whereas only 41.7% of the control animals had a better response. That rose to 71.4% for the treated animals, indicating a much better response. They also then stained the optic nerve of the animals, and the staining indicated that there was a lot less damage in the treated animals. So these are sort of cross-sectional stains, I guess. Here in G, the bar graphs of, I guess, that staining, so the nuclei area and the IBA1 plus area was much lower in the treated animals than in the control animals. So you can see the gray bars and the blue bars, and that reached statistical significance and is a good thing, as it indicates less inflammation and less damage. And on the right here, once again, sort of staining, IJ and LM, and basically this was showing the myelin content. And basically neurons or axons are surrounded by myelin, the sort of protective insulator, just like you have plastic coating around an electric wire. And more is better. And as you can see here on both of these bar graphs, K and N, the blue bars are higher than the gray bars. And this one was sort of on the verge of statistical significance. And remember, statistical significance is a p-value of below 0.05, so it sort of hit that, whereas in N here, it did reach that. And in I to K here, this shows the amount of myelin. So myelin is a protective coating around neurons and axons, the same way that an electric wire has sort of that plastic insulator around it. And more is better as that sort of gets damaged in multiple sclerosis. And we can see here that the blue bar of the treated animals is much higher than the gray bar of the control animals. However, it sort of just reached statistical significance with a p-value of 0.05, and statistical significance, as a reminder, is a p-value below 0.05. So it sort of just reached it. And on the right here, L, M, and N is showing neuronal survival. And once again, the blue bar is higher than the gray bar. And this time, it did reach fully statistical significance as denoted by this star here. And lastly, they also looked at that senescent marker BCL and the staining for that. I guess these images show that. And in the IBA1 plus area, which is the specific area of the optic nerve they were looking at, there was a statistically significant decrease in that senescent cell marker. However, that wasn't in the sort of overall optic nerve area, which is supposedly a good thing. So overall, it appears that using senolytics is positive for this model of multiple sclerosis. And although a lot further work is needed, this work provides an avenue of hope for treating multiple sclerosis, which currently is incurable. Now on to our second Canadian study and final paper. In this study, researchers from the University of British Columbia wanted to find out how phytoestrogens target the hallmarks of aging. The phytoestrogens they chose were resveratrol and pterostilbene, using chlorogenic acid from coffee as a control. However, they found out that resveratrol and chlorogenic acid had no effect at the concentrations they used. So this paper is only on pterostilbene. Pterostilbene is one of the main active ingredients found in blueberries and has been shown to have numerous health benefits. This study was done in rats, and they were fed pterostilbene for 20 days before their blood was sampled. They started off by seeing how gene expression changed. Without going into too much detail, they found that numerous genes were either upregulated or downregulated. Looking at C here, the blue shows downregulated genes and the pink shows upregulated genes. These were the genes which showed the biggest change. And for the two pink upregulated genes, for interleukin-4 production and interleukin-10 production, they represent anti-inflammatory molecules, so upregulation is a good thing. For the blue downregulated genes, they mainly involved pro-inflammatory interferon-mediated signaling pathways, whose downregulation is also a good thing. They then showed how more specific genes target the hallmarks of aging. At the top here, you can see the top differentially expressed coding genes, with the turquoise being upregulated and the purple being downregulated. And here's a list of those top 60 genes. And below is a descriptive summary of gene function and how they might impact longevity. So you can see it says increased longevity here, and the various genes on the right, with the purple being, again, downregulated, turquoise being the upregulated genes. And they go from regulation of lipid metabolism, which is atherosclerotic protection, so protection of the arteries, increased dietary restriction, usually that is a good thing, increased autophagy, again the recycling mechanism in the cells, which again we want more of, increased mitochondrial biogenesis or homeostasis, increased genomic stability, decreased ribosomal biogenesis, regulation of transcription and intercellular interaction, regulation of stress response, bacteriophagocytosis, decreased inflammatory immune response, regulation of DNA damage repair, and chromatin regulation. Needless to say, impacting these various hallmarks is a positive thing. Then they looked a bit closer at a few of these genes. And in each of these graphs of 8 different genes, the white boxes represent the expression in the control animals, and the blue boxes represent the expression in the pterostilbene supplemented animals. The first 4, from IRF7 to IFI27, affect the inflammatory immune response. And generally speaking, lower is better, and for each of these, there was a statistically significant decrease as denoted by the star. LGALS3BP is associated with the regulation of intercellular interaction, and the expression of this gene was found to be lower in men who lived longer. So the fact that pterostilbene lowered it is also a good thing, and again, it was statistically significant. RTP4 is associated with the stress response, and again, lower should be better. SLC7A5 is associated with dietary restriction, and as we should know, dietary restriction is also considered a good thing for longevity. However, this reduction did not reach statistical significance. The same goes for PLTP. However, higher levels are considered beneficial, so the researchers weren't sure what was going on here. So far, all these experiments were done in rats. However, the researchers also wanted to see how these genes changed in humans, starting by looking at the differences between younger and older humans, in this case, 46-year-olds and 68-year-olds. And they used two different data sets for each of these genes, though the order has been changed from the previous figure. And overall, the results were fairly inconclusive. For although a number of them seemed to show an increase, as you can sort of see here and here, for only one of the genes, OAS1, and one of the data sets was there a statistically significant difference, as you can see by the star here for this data set. They also looked at the gene expression differences in a couple of disease states. Firstly, steatosis, which is fatty liver disease, and also non-alcoholic steatohepatitis, a more severe form of non-alcoholic fatty liver disease. And first, they looked at the 4 genes associated with inflammatory immune response. And the green are normal people, the orangey pink are the steatosis, and the blue are NASH, or the extreme non-alcoholic fatty liver disease. And also they used a number of different data sets. You can see along here with the number of people in each of the data sets also mentioned, also a couple over here. And for several of these, there were statistically significant differences between healthy people and those with either steatosis or NASH, as you can see here on the left. So there's a statistically significant difference both for steatosis and NASH. Here for this one only for steatosis, for this one only NASH. And again here, these are 2 statistically significant increases for people with NASH. Further down are IRF7. This one data set had a statistically significant difference. Over here, however, there seemed to be a decrease. OAS1 over here also showed a statistically significant increase, as it did over here as well, but not for OAS2. Looking at the other genes, we can see that a number of them did show significant differences, again denoted by the stars on some of these data sets. Yeah, I'm not going into this in too much detail, lest we get lost amongst the weeds. All in all, the authors conclude that the pterostilbene target genes are enriched with functions that are tightly connected to slowing down aging and increasing lifespan. So it does appear that eating blueberries is a good thing, at least if you are a male rat, that is. And that marks the end of the penultimate longevity review of the year. Please don't forget to like this video, subscribe to our channel, and take a look at our new online store. So until next month, goodbye.